Exactly. We are really excited to introduce Professor Roland Zengerle. He's a professor for MEMS applications at the University of Freiburg and also an executive board member of Hahn-Schickard. And it seems like in your title, we are reading about molecular diagnostics and the Point-of-Nee. I think Jerome just mentioned that in his presentation as well.
Yep.
So I feel it's like the perfect segue to your presentation now.
Well, thank you very much for this kind introduction, and also Sir Jerome do interesting presentation. I had also one additional question posted. By the way, you mentioned that Q has 230,000 installations, which is 5 times more than Cepheid. I'm surprised to hear that, and maybe you can answer it on the chat. Okay, so I share my screen, and I'm now going to talk about molecular diagnostics at the point of need, and I will start with introducing myself. So I'm speaking today for Hahn-Schickard. Hahn-Schickard is a non-profit organization, about 260 employees, 4 different locations in the southwest of Germany: Stuttgart, Villingen, Freiburg, and Ulm. And I'm based in Freiburg, and I'm also associated with the University of Freiburg, as you already heard. So this is the outline of my talk, and I'm talking a bit about my personal past and future. And I start with the 90s, which was component-driven thinking, and I continue with the new millennium, talking about point-of-care diagnostics and some aspects about the future. And as some others also, I started working with micropumps in the 90s, and I think this was the first early hype in MEMS and in microfluidics. And we thought that we could do everything with micropumps. And this is the micropump I worked on. It's 4 pieces stacked on top of each other, 2 valves, 1 diaphragm. And it looks quite simple, but it turned out to be much more complex than I ever thought. Yeah. And really, it's interesting to see that others like Martin Richter, who is also on the meeting, and Bartels Mikrotechnik, they continued to work on micropumps until now. For me, it was too complex at that time. And I also worked on microvalves, by the way, electrostatically controllable microvalves for pneumatics. The valve could control 10 atmospheres of gas, but it was much too expensive to fabricate. And so the only application for that kind of device was a space mission, where we had 28 of those valves flying to a comet. And they arrived 10 years later in 2014, and they did some measurements on the comet Chury. And so this was the early days in my personal career. And my personal conclusion after the first, let's say, 5 to 10 years was, for me, it was much more important to understand the application first, and then think about the most simple solution that can fulfill the application. And in terms of microdosage and nanoliter dosage, it was a device as simple as this. So we only used a tube, polymer tube. We squeeze it by a piezo-stack actuator, and we can easily generate nanoliter droplets out of it. And all that is independent on viscosity. And it was just a symbol that thinking on it and the application first, and then try to come up with a microfluidic solution. This was guiding me for the rest of my career. So I will continue with the new millennium and with this application-driven thinking. And we targeted applications like point-of-care diagnostics or next-generation sequencing. And I will give an introduction to the point-of-care diagnostics. So what does it actually mean? It means that you look for viruses or pathogen in a human body. And as you all know, this is the SARS-CoV virus. And we now have 2 approaches. We have an antigen test. The antigen test is looking for proteins, as, for example, so spikes on the surface of the virus. But for this kind of antigen test, you don't have an amplification that is possible. That means it's less sensitivity, there's a lower sensitivity, and that means that you need about 100,000 viruses to be able to detect an infection, 100,000 viruses. In comparison, for a PCR test, you go for the RNA of the virus. Of course, you need a more complex biochemical process. You have to translate to transcript the RNA into complementary DNA. Then you can start a PCR amplification. You get about 1 billion of copies of this DNA molecule, and this enables you to detect the virus at a limit of detection of about 100 viruses, or maybe even 20 viruses in a swab. Yeah. And this is what I'm going to talk about now. If you look in centralized facilities, you have instruments like this. They can do about 10,000 samples a day for a PCR test. And I don't go too much more into the details here. Just want to mention that the total capacity in Germany is about 2 million. Oh no, it's I guess 2 million PCR tests a day. And the challenge that we are focusing, employing microfluidics, is to shrink all that to a device like this, which is a desktop device and a disposable cartridge. And this device should do the test in something like 30 to 40 minutes, at least less than an hour. And the settings are emergency settings in doctors' offices. And now there are, of course, some challenges associated with that. So systems has to be easy and robust to use. You need to do sample to answer systems. Yeah, that means you add the sample, push the button and get out the result. You need to have the same requirement as in centralized facilities. No one would accept different results coming out in a point of care test. And you have to be able to manage quite complex biochemical assays. It's still cost sensitive, and your gain is the shorter time of result. And how to do that, how to tackle this challenge? This is the way we usually do it. So we break down the complex workflow into individual blocks like sampling, lysis of the virus, binding the RNA, washing, elution, liquid transfer, and do a PCR reaction. And we call those building blocks the process chains. Most of the biochemical assays in any field of medicine you can break down in process change like this. But of course, if you do sampling, for example, you can still break it down in more basic steps, like you need a sampling device, you have to take up the sample, you transfer it to the cartridge, you inject it into the cartridge. Or for example, if you lyse the virus, you need, for a chemical lysis, you need reagents, need to do a mixing, need to do incubation, and so on and so on. I don't go into the details, but all those basic fluidic operations we call the fluidic unit operations. And you see that some operations like mixing, they happen quite often, or valving, they happen quite often. So you need mixing, valving all the time. You also need pumping, let's say, or sample transfer, fluid transfer, and so on. So we do a rearrangement of those building blocks. And we realized that in order to do a lab on a chip system or a point of care test system, you need to be able to perform all those fluidic steps, bead handling, incubation, aliquoting, mixing, metering, and so on. And you immediately realize it's a system integration challenge. It's not a component challenge, finding a mixer, finding a pump, it's a system integration challenge. The most important aspect that you need to have a solution for all those unit operations on the left-hand side is usually, but it's more important that they are easily combinable, yeah, in a low-cost fabrication technology, ideally in a monolithic fashion to be cheap and simple. And that's it. And in the next slides, I will present you an example how this can be done with just 2 layers of fluidics, one microstructured layer functionalized with reagents, and one cover which seals all the chip. So simplicity is the key. Before I go into the detail of what we did, I just want to mention that at the moment, there are quite many point-of-care platforms for decentralized molecular testing available now. Cephite, the market leader, and also other instruments that you see here. And they all employ microfluidics. So microfluidics is everywhere, in my opinion, if it's about point-of-care diagnostics. So I will continue to to present you our solution. And our solution was a centrifugal microfluidic platform. And what does it mean? Well, we have a device which is here. We have a cartridge with it there. We load the cartridge, and then the cartridge is oscillated, is spinning. Yeah, and you see the spinning protocol here. This is the spinning frequency over time. And the second axis, this is the temperature in the cartridge. And by controlling the spinning frequency, you can control the processes. For example, releasing of the buffers, do mixing, do washing, and do all the processes that we need to do. I will give you some examples. Why have we done this? This gives us the most easiest cartridge. So we do not need any active pumps or tubings. So we have easy interfaces. We don't have issues with gas bubbles, as we have already seen again in the experiment, because gas is easily to be separated with liquids on a centrifuge. Yeah? It has a much lower density, and it's easy to use at the point of need. I will show it later. So I give you some examples how we implement some functions, and important functions, I guess Holger Becker talked about this morning, is reagent storage and release. So how do we store liquid reagents? We pack liquid reagents in those stick packs that you know from ketchup bags, for example. Yeah, we fill it in. The whole stick pack comes from a polymer roll. It's sealed here and it's sealed there. And we fill in the reagents and we cut the stick packs and we load the stick packs to the cartridge. So this is how we store the reagents. And now by centrifugation, by spinning, you can easily generate a pressure acting on the seal of a stick pack, and the pressure peels the seal and releases the liquid into the cartridge. It's a very simple process. I shouldn't say simple, but it's an elegant process, I think, and it allows you to do long-term storage of reagents in cartridges. I will give you one more example of a process. It's liquid transport. So this is the center of a rotation of a cartridge. This is the outer rim. If you have liquid here and you want to move it, just start spinning. And by centrifugal forces, of course, the liquid will move from the inner part of the disc to the outer part of the cartridge. Okay, one key problem was how to get the liquid back to the center. And this is also illustrated here. So the liquid was moving towards outer part of the cartridge. This was a closed cavity. Air is captured in the closed cavity. So you build up an air pressure here. And if you now stop spinning, the pressure releases and liquid goes out. And if you have two channels, one with a high resistance and another one with a low resistance, you can get all the liquid back. Well, not all, in this case, 90%, but you can get back the liquid to the center of the cartridge. Okay, so you have a pump pumping liquid from the outer part to the inner part, and the pump does only cost you one channel. Yeah. Now, how do we do many tests at the same time or many targets at the same time? Typically, we do geometric multiplexing in our cartridge. That means that we have here cavities. They're all loaded with different molecules, so-called primers and probes. And in every cavity, we amplify a different DNA signature, looking for a different analyte. For example, SARS virus or an antibiotic-resistant bacteria or whatever. The only thing we have to do is do thermocycling. That means changing the temperature between 60 degrees and 90 degrees and monitoring the fluorescent light coming out. If you have a target detected, then you will see fluorescent light coming out. If you count the cycles that you need in order to see the fluorescent light, you get the CT value of the PCR reaction that you probably know from your SARS-CoV test. And this is very sensitive. So the limit of detection in principle is about 1 molecule per cavity. And that's reality. You find any molecule if it's present in such a cavity. Now, this is the overall sketch of the cartridge. It's one microstructured part. It can be fabricated in injection molding or thermoforming. Then we do functionalization. That means we bring all these liquid reagents into the cartridge, different buffers. We also bring in magnetic beads, for example, that increase the surface that we need for our biochemical reactions. We bring certain reagents for the PCR reaction, like lyophilizates, and we add primers and probes in those cavities that allows us to do a different test in any of those cavities. Yeah? And then you put the seal on top of it, and that's it. It's only 2 layers that are needed for doing this product. We created a spin-off company called SpinDiag. The spin-off company was created in 2016, unfortunately not 2009, because when the pandemic came, they did not have the product on the market, but within 6 months from the pandemic, in November 2020, they were able to bring the first product to the market, which was a SARS-CoV-2 test. The machine is doing a test in 50 minutes, 2 tests in parallel. That means that you can feed 2 cartridges at the same time to the instrument. And the sensitivity is about 26 copies, virus copies in a swab that you add here as a sample into the cartridge. So it's quite simple. The key is to have the complexity in the instrument. So this is the spinning motor in the instrument that spins that table. The table also has heating zones that can heat up fast and cool down also fast by Peltier elements. So they can do a PCR in 20 minutes. And the sample prep, which comes before the PCR, it adds up to this overall time of about 40 to 50 minutes. It's quite easy to control or to operate. Again, fabrication is also a key. The whole test is made out of 5 pieces. Actually, so far I only told about 2. It's the microstructured substrate. It's a polymer film for covering the substrate. Then you need a cover for the label, and you need the cover cap for the sample port, and that's it. It's only 5 pieces. And at the moment, of course, it's a platform. So if you have one cartridge for SARS-CoV-2, you can also do other tests like antibiotic-resistant bacteria, which is coming out now, or the respiratory diseases. This detects also flu and SARS-CoV-2 and several other pathogens or viruses. They're all now close to the market release. And the system is at the moment in 260 German hospitals present. It's only available in Germany, but international scaling is now the next step to come. Overall, I talked about nanoliter dispensing and centrifugal microfluidics. There are several products that came out of this kind of research. The nano dispensing of biofluidics in 2005. Then a single cell dispensing instrument from Cytina, now PICO solutions. And we also brought out a Sievers Eclipse test for bacterial endotoxin testing with the company Sievers or SpinDiag Ronda. And the strength of the platform means that you can also use the same techniques for also other applications. And so there are 2 more spin-off companies created in the last 2 years. Diagnostics is looking for dermatology applications, and Anderson-Hauser Biosense is looking for all industrial applications for in water, food, and beverages, also in wastewater, where the molecular testing is interesting to do. You can, for example, also monitor SARS-CoV-2 in wastewater treatment and can see outbreaks of cities before the people really feel ill. Now, this is the present, and I will also give you some, more or less 2 examples on what I think is the future of microfluidics and what we can do in the next decades. And there are huge challenges left. So if you think on a human body, for example, you have about 20,000 genes in your body, about 1,300 proteins that are used in R&D for investigating illness. And if you look to the diagnostic systems that are available, so typically if you are sick, you have targets, biomarkers that have a high concentration of 1 nanomolar. And but you also have, if you are ill, if you are healthy, you have much less concentration of these proteins in your body. And the present state of the art in vitro diagnostics, they just look to about 200 different proteins, more or less at high concentrations. Yeah. So the future is to go into these segments of lower concentrations, and in order to look at more biomarkers. So the humans have 20,000 genes, about 100,000 proteins that could be of interest, and about 800,000 protein-protein interactions that also could make a difference, for example. Yeah. And now the question is how to deal with these lower concentrations and with a multitude of more parameters. And I will give you a very first example what we do in this regard. It's a new kind of assay that employs microfluidics, and we call that assay protein interaction coupling technology. And what is the mission of that technology? We want to detect proteins. This is a protein in yellow. This is a blue protein, let's say yellow and blue. And this is the interaction of both proteins. And we want to detect this in individual cells by technology that employs barcoding the proteins with DNA, let's say a blue label and a yellow label. And I will illustrate in the next slide how we do that. First, I have to explain PCR and digital PCR. So the standard PCR was for detection of DNA. And you do it by amplification of the DNA, and then you detect it. In the early days, it was with gels, and nowadays all people do qPCR, which is a quantitative method. That means, again, you are detecting DNA, and you detect do an amplification. You detect the fluorescent signal coming out of the amplification, and then you get fluorescent characteristics like this. The number of cycles when the signal gets up here is a measure of the amount of DNA that you have in your sample. Yeah? This is low concentration. This is high concentration of DNA. And there's a new technique called digital PCR that does compartmentalization of the analyte. That means you distribute your analyte in 10,000s of droplets, and then you have droplets that are empty, and you have droplets that cover or just contain one, seldom two DNA molecules. And if you now do amplification in droplets, you get signals like this. So you have many empty droplets and you have a few colored droplets. And now you can count the number of molecules that were in your sample. This is a digital PCR. And in digital PCR, you have instruments, for example, from QIAGEN, and you do an analysis by just counting the number of positive droplets. And digital PCR gives you a higher amplification yield and a higher robustness. You will start with individual molecules. It gives you a higher sensitivity because you have less cross-contaminations in those droplets, and it's a proven technology. So how can we detect proteins by digital PCR? This is now illustrated on this slide. So we have two proteins, yellow and blue, and we want to know if they are individual proteins or if they react with each other. Then we need antibodies, let's say a blue antibody sticking to the blue protein, a yellow antibody sticking to the yellow antibody protein, and both antibodies are also labeled with DNA labels. So this is just done in solution, mixing everything together. It's quite mixing, only mixing, nothing else. Then we do a dilution and we distribute the analyte to the droplets, and then we have droplets containing individual proteins, or we have combinations of proteins. Then we do an amplification of the DNA label. That means we get yellow, blue, and green droplets. And then we have to do an analysis by a standard digital PCR instrument. So we have the green fraction, the blue fraction, and the yellow fraction. And this is the amount of protein interactions. And this is the amount of empty droplets. And that way you can count individual proteins inside samples, or even inside one cell based on this technology. And this is now commercially available since a few days. It's sold by QIAGEN at the moment. And PICO can do many kinds of different assays. So for example, you can do quantification of one protein by this technique. You just need 2 different antibodies for one protein. But you can also detect post-translational modifications, like for example, a phosphorylation of a protein by having 2 antibodies against it. You can detect protein-protein interaction coupling, and you can by having 2 different antibodies, or you can also detect modifications of the individual proteins that you can do with the same technique. So the experiment looks different. The outcome of the experiment, you have pictures like this. If you zoom in, have pictures like that, and you count positive droplets. And there's a software for doing this, and you can have really quantitative results by this technique. So this was one first example. For the future, I will spend a few more minutes for a second example, which is nanopore-based molecular diagnostics. The slides are from my colleague Felix from Stetten, and the thinking is the following. You know that, for example, Oxford Nanopores, they brought this minion device on the market. It has the size of a USB stick, more or less, and it can do sequencing by just feeding the DNA through nanopores. Yeah? And there are also ions going through the nanopore, and all the time when a nucleotide blocks the nanopore, the current changes, and by those current signatures, you can analyze the DNA which is going through here. And we think that this in the future will also be used for protein analysis. And there is a multitude of options that you can do. For example, you can work with bacterial nanopores. You can use with also solid state nanopores. And you can do different measurement principles. You can, for example, do the electronic measurement of ion currents going through the nanopores. You can do optical measurements. You can do combine optical with ion current measurements. So there's a multitude of different alternatives. But of course, the detection of proteins is quite complex. So you get, if you see the pore here and you monitor the current, you have current signatures like this here. And you have, first of all, do denoising of all that signals that you get. Then you detect the spikes, then you do feature extraction, and later on you do an analyte, calculate the analyte properties. And there are quite promising first results about this. Yeah, this is recently also not from our group, but from a different group, a science paper about protein analysis through nanopores. And we think that this also will have a great future. So, currently, state of the art is DNA and RNA sequencing for proteins through nanopores, or through of DNA and RNA sequencing through nanopores is state of the art. In the future will be protein, peptides, recognition of small molecules, also sequencing of carbohydrates, and they all are relevant for our health status. This brings me to the summary, and yeah, the pandemic changed a lot in public awareness of rapid diagnostics. Everyone understands now that having rapid results is interesting and relevant. And actually, I just put some devices here, GenMark or Bosch Vivalytics or Spindia Gronda. They're all based on microfluidics. Yeah, GenMark on electrowetting. Bosch is a pressure-driven principle. Actually, they are employing an elastomer membrane, almost like PDMS, and Spindia centrifugal microfluidics. So there is no way to do point-of-care testing without having microfluidic principles involved. From the lab-on-a-chip perspective, sample-to-answer analysis at the point of need is now a reality, and the microfluidic platforms are the key to success. Only on a platform-based approach, you can easily bring new applications on the same cartridge, or you can rapidly change from one test to develop a new test, as it was the case for SARS-CoV-2 pandemic. And there's, of course, a huge variety of microfluidic approaches. There is not one concept fitting everything. clearly better than all the others. So there is a multitude of different approaches. And nevertheless, it's wise to know about the multitude of tools and options when you tackle new challenges. And an outlook, key challenges for the near future are acceleration of time to result. So 45 minutes is still long. You need to go down to 15 minutes or 20 minutes without losing quality. Multiplexing will be an important part. So technically, we can do 30 to 50 assays on one cartridge at a time. And I think a great future will also be novel tools. We will have novel tools for diagnostics in the future. I gave two examples to detect hundreds of proteins and protein interactions by the PICO technique, or the nanopores as detectors in diagnostics. And there are, of course, some engineering challenges left. Microfluidic platforms for in vitro diagnostics at home is still a challenge, and reducing the costs for tests is always a challenge. And this brings me to the end. Thank you for your attention.
Thank you very much, Professor Zengerle. We also had two questions while you were in your talk, and one was regarding the spin diag. What cost per test do you estimate?
The test is sold, I guess this is known or is public, around 30 euros. It depends how many tests you buy, and one test is typically 30 euros or below.
Okay.
And fabrication of those cartridges is in the euro range, so it's not two-digit euro range.
Okay. And then the other question is there an open source system for centrifugal microfluidics which can be used by researchers?
Interesting question. Actually, we support any researchers, and we can support also these centrifugal instruments, though they are free to use to everyone, and just contact me.
Okay.